A very good afternoon to all of you again. Have a nice and safe day. Okay. In this video tutorial, we are going to discuss about another short and important topic in biology, that is about T cell activation. The immunological brain, it is very much important for body immunity. Okay. So, let us discuss in detail what are the molecular events which occur for activating T cells. The dendritic cells are brain of immune system and T cells are heart of immune system. Why? Because if we compare, if we think about T cell, the T cell produces a particular group of Th cell, helper T cell. The helper T cell induces to produce more cellular immunity, and also it activates the B cell into plasma cell. More T cell, it produces more T cell and convert B cell to plasma cell. The plasma cell produces antibody, plasma cell produces antibody, and T cell directly produces cellular immunity. So, the immune system can be stimulated by T cell. In spite of T cell, B cell activation is not possible. Clear? So, today's class will discuss about the activation of T cell. What are the various stimuli needed for T cell activation and the mechanism of T cell activations in immunology. Clear? So, let us discuss. My dear students, there are two major type of T cell: CD8, CD4 cells. Okay. The CD8 marker T cells are cytotoxic T cell, or we can call it killer T cell. The CD4 T cell are helper T cell. These are helper T cell. The cytotoxic T cell, what does it do? The cytotoxic T cell, it directly kill the cell by producing certain chemical. It kill the cell. A specific cell is killed by the cytotoxic cell. What the function of helper T cell? The helper T cell activate B cell as well as other T cells. Clear? So, these are important. The mechanism of activation of these two cells are also similar. A same pattern of stimulating effect has been induced by the macrophages, antigen presenting cell. So, we will discuss about CD4 T cell, helper T cell, because it is the prime cell for activation of whole immune system of body. Clear? So, as we know, what the antigen presenting cells, the most important antigen presenting cell is dendritic cell. Okay, these are called as brain of immune system. Macrophages, dendritic cell, B cell, they are very important antigen presenting cell. These have APC, these have MHC2. Okay. So, suppose this is one of the macrophages. This is a macrophage. What happens? At first, when any pathogen enter into body, macrophage, suppose this is the pathogen, we will take it This is the pathogen. What happens when pathogen invade into body? Immediately it is taken up by this, engulfed by this phagocytic cell. It is engulfed by the phagocytic cell. It is degraded. It is degraded to produce exogenous proteins. What happens then? These exogenous protein remain at its surface because here these are antigen presenting cell. These antigen presenting cell represent through MHC2. Suppose this is the protein fragment, they are represented here. This is MHC2. Okay. So MHC2 represent. Then what happened? This MHC2 show it to T cell. Suppose this is one of the T cell. T cell. This T cell is in inactive stage. We are discussing this CD4TH, that is the helper T cell here. What happens? It is the helper T cell. Helper T cell here. Okay. How it is activated? First of all, there is a receptor on the surface of this helper T cell called as TCR, T cell receptor. This T cell receptor first interact with MHC2, which is very important phenomenon. The first phenomenon, TCR with MHC2. MHC2 already it consists of the pathogen, exogenous agent, proteinous component that is degraded inside it, so that it shows the protein that these are the protein to activate the T cell. Okay. So there is a basic interaction between MHC2 and T cell receptor, and it is very important for activating the T cell. First and prime essential, the interaction of these two agents is associated with certain co-stimulatory effect. What happen? The co-stimulatory activities are B7 of macrophages interact with CD28. There is another. This is the macrophages consist of B7. It interact with CD28 receptor of T cell. CD28 कहाँ पे होगा? ये T cell में होगा. This became the CD28. So B7 with CD28. It is a accessory event, not important like it. Okay. What happens then? CD40 react with CD40 lesion of T cell. T cell में क्या होगा? CD40 lesion होगा. CD40 lesion here. This is CD40 lesion. This CD40 lesion react with CD40. Where it is present? CD40 is found in, it is found in macrophages. Okay. So second, CD40 with CD40 lesion. It is another co-stimulatory effect. Clear? After that, these interactions are checked by CD4, which is present in this CD4 receptor present in T cell. It is another important receptor, another color here. This is a CD4 receptor. Then the second event, CD4 receptor check all these reactions. Okay, CD4 make all these three interactions perfectly interacting zones are regulated by CD4. Okay, so now the T cell properly engaged binding with macrophages. Okay, T cell is properly engaged for the process of activation. What happened then? Now the chemical phase appears. Chemical phase, this is the molecular phase. What happens in the chemical phase? Let's write it. First, in the chemical phase, this cell means the macrophage cell started producing interleukin. This interleukin which is released here is interleukin 12. Remember it. This interleukin 12 is produced by it. Interleukin 12, what happened? This is the pathway of secreting interleukin 12. There is a interleukin 12 receptor found in T cell. Here, this is a receptor, IL-12 receptor. It received the interleukin 12. Clear? What happened? Immediately, the T cell produces interleukin 2. Okay. Immediately, this T cell produces interleukin 2 with response to interleukin 12. What happened here? From this helper T cell, interleukin 2 released. Okay, it is one of the endogenous substance, not exogenous. When interleukin 2 is released, immediately the T cell produces interleukin 2 receptor. Sorry, this is interleukin 2 receptor. This interleukin 2 bind here and again goes to the T cell. Remember it. When the material is secreted from this cell and act in the same cell, it acts in the same cell. This pathway is called as autocrine activation. It is called as self activation or autocrine activation. So the T cells get activated by interleukin 2. So it is all about the process of activating T cell. The process of activating helper T cell is similar with activating killer T cell. So thank you all. We will discuss B cell activation in another video. Okay.